All right, so welcome to another module for the course, one of our last modules actually, since we're getting close to the end of the semester. And in this module, we're going to kind of shift from what we've been talking about in the semester in terms of, organic molecules, you know, um, chemical processes, theory, all of that stuff. And we're going to kind of bring or come back down to the biological hierarchy. We're going to focus on populations and communities specifically. So we've talked about the diversity, how the diversity arose. Now we're going to look at how that diversity interacts with one another on a regular basis, and how they can influence one another. Because like we see in this image here, one ecosystem is made up of multiple different species, right? And here you have a lot of, larger organisms like your birds and your river otters and your quail and your minks and your red fox, deer, etc. Um, but then there's also microbes, and you can see the fungi, and then there's your abiotic factors. And so we're going to look at how all of these things can influence community and population dynamics. And looking and learning about these interactions is part of the field of ecology. All right. And so whenever you're studying communities, you're referred to as a community ecologist. And so what is ecology anyway? Right. And so if we look at the following list of terms below, right? Just take a minute and think about, out of all of these terms, which of these are part of the field of ecology? So what do you think? All right. And so in looking at this, hopefully you thought, okay, at least the living factors are part of ecology, right? Because that's your organisms. Then you have like your environment, your organism, which is part of living. You have your soil, your weather, your population, community, planet, right? And so in reality, all of these terms are related to ecology, and they're important for ecology. Okay, so now that we've said that all of these terms are related to ecology and important for ecology, which of these are biotic and abiotic factors? Okay. You remember those terms from earlier modules? So go ahead and pause the video, take a minute to think about which of these are living and non-living factors. All right, so hopefully living and non-living is obvious, right? So living's biotic, non-living's abiotic. Your environment, your weather, your soil, and if you're thinking about the planet just as in terms of the planet and not the organisms that live on it, then those are your abiotic factors, your non-living factors, right? Whereas your organism and the populations and the communities are your biotic factors. Now, the planet and the soils can have biotic components inside of them or on them, like the soil can have living microbes and the planet has all of the living organisms on it. And so in those, it's just looking at what level or scale your study is looking at. And and speaking of those levels and scales, what exactly are levels of ecological studies? And these levels are really dependent on the questions that we want to ask about specific organisms or specific populations or communities, right? And so one of the smaller scales is, let's just have a question about a specific organism. So maybe, how is a lizard influenced by temperature? So how is this lizard influenced by temperature? Okay. Or we could say all of the individuals of a given species. Right. So let's look at a population level question. Right. So does slope affect the behavior of this population? Or slope and aspect, I should say. And so slope and aspect is basically saying, how steep are cliffs, right? How steep is the slope of the landscape? And then the aspect is the direction in which the sunlight hits the landscape. So north facing slopes versus south facing slopes. And we know that these aspects actually have different levels of radiation. So for example, north facing slopes are much cooler than south facing slopes. And so how does that affect the behavior of the population of all of these individual lizards? Do you find them more on south facing slopes? Do you find them more on north facing slopes? And or are there equal amounts for this population? And so here we've moved from a single lizard to a population level, and then you can keep going, larger and larger in this question. And so we may ask about all of the species in a specific area. So does the presence of one species affect the presence of others? So for example, if there's a wolf present in this habitat, how does it affect the elk population or the cottonwood population or the willow population? And so all of those are very important questions, and they all range from different levels. So here we're really at a local level where we're looking at an individual lizard, and we can look at this individual lizard in a lab. We don't even have to look at it in its natural environment, but we can also do that. And then we go up to its population, and then here we're looking at a community. And then we could even go larger and larger, and we can look at regional or continental influences. And so there are a variety of levels of ecological study. And even though you're going up in scale, you can still only focus on one species, or you can focus on multiple species. And so that change in level and scale really is relegated by the type of question that we're asking as ecologists, and and the hypothesis that we're trying to understand. And so just to kind of review, right, because we are talking about populations, community ecology, population demographics, and all of those things, we want to review and make sure we know exactly what a population is. So if you remember from our first two lectures, right, a population is just a group of individuals from the same species, and they live in the same location, right? So for an example, if we look at humans, we have human populations all over the globe. Even though humans are from the same species, we can divide ourselves up into populations, right? So you have like your Merced population, you have your San Francisco Bay population, or you can even get larger. Populations are defined on the scale. And so populations, say we can have, instead of just the Merced and San Francisco population, right, break it up in cities in California, you can have your California population, or you can have your Texas population, right? So you can break it up into states. And so again, your question determines the scale of the population that you're looking at. And now populations, they'll change and they'll fluctuate in their sizes. And so population sizes can change just based off of simple environmental variation, right? So let's say you have like seasonal weather. So like in California, we have wet winters and dry summers. So maybe your populations change in relation to that. Or maybe there's yearly changes, right? So from year to year, your population will change. Or maybe it's, decadal changes, or they change across space, right? And so the environment and those abiotic factors can really control population sizes. Natural disasters can relegate population sizes. So let's say you have a volcanic eruption that's going to get rid of individuals in a population due to mortality, right? Floods, hurricanes, big weather events. Those are going to affect population sizes. And what's interesting about natural disasters is evolution does not play a role, or I should say fitness does not play a role in who gets selected to live or not. It's random. And so that's kind of an important concept that comes from natural disasters. And then we also have species interactions, so like competition. So let's say you have one plant that's a really good competitor and it outcompetes other plants. That one plant can actually prevent other plants from living in that environment. And so they can competitively exclude other species from that community through those species interactions. And now that we've kind of defined populations and talked about what influences population size, we can characterize populations based off of its size, right? So its number of individuals in the species, or we can characterize it based off of population density. Population density is basically the number of species in a given unit area. So for example, you can have 50 deer per kilometer squared, or you can have like 30 plant individuals per meter squared. Okay, and so that's a population density, and we have those numbers for humans, right? So like Merced's population density may be, 200 people per acre or something, right? And so population density is pretty important because it can affect species interactions. Now, you could have a large population size, but if those individuals are over a really large area, their density is going to be low. Whereas if you have a large population size and they're over a small area, their density is going to be high, right? So density can tell you some information on species interactions, right? How often species are coming into contact with one another or individuals from that same species, which can give you information on resource competition, right? If you're pretty dense, then you're going to be competing for space. You're going to be competing for the similar resources. Whereas if your population density is lower, there's much more area per individual in terms of resource allocation. Also, mating success, right? If you have a low density, it may be very hard to find a mate. But if there's a high density and you're bumping into individuals on a regular basis, it makes it easier to find a mate, and therefore mating success can actually increase. And so smaller organisms generally have denser populations, right? And so in general, if you look at the mass of an organism and the size of that population, your smaller organisms, right, or I should say the density of that population, your smaller organisms have a much larger density compared to larger organisms, right? You can see that's fairly linear, pretty good relationship there. Now, the way we measure population size and density is through a couple of different ways. And the way that you will measure those organisms depends on the species that you're looking at. So for example, if you're studying an organism that is sessile, meaning that it doesn't move, so like coral or plants, you can use a quadrat. And so we have two quadrats pictured here. Generally, these quadrats are 1 meter by 1 meter square. And so that gives you a square meter of area inside, right? And so you can count the number of individuals in that square meter, and you can extrapolate, right? So you can use the number in that square meter and you can expand it and predict the total population size or density. These quadrats can be made of different materials. It could be wood, plastic, metal. And the key thing is you want to randomly place them on the ground. So if you just take a quadrat and you walk over and you're like, oh, there's a lot of individuals here, and you place it down, then your counts are going to be biased, right? And so what researchers will do is they can basically set the study area up into a grid, and they can randomly choose numbered grids to sample from. And you can sample, multiple grids, right? So let's say if there's like 200 grids, you're not going to sample all 200. Maybe you'll sample like 20 of those grids, and you can take the average, and then you can extrapolate that. And there's also other formulas that you can use to extrapolate. And so you complete that multiple times in a habitat to basically get a more accurate estimate of population size and density. Because what if you select one grid and maybe the environmental conditions in that grid, like plants. So if this is like a sandy area beside, like a nutrient rich area, your counts are going to be different, but it's still part of that habitat. And so that's why you want to make sure you sample multiple grids so that you can make sure you're getting an accurate measure of the population size and density of the species in that area. You can do the same thing with corals as well, and other marine organisms that are sessile, so like kelp, that sort of thing, where they'll just go in and randomly put quadrats down and sample those populations. For your mobile organisms, especially smaller species, but we can do this with larger species as well. Larger species, we typically will do like tags, so like satellite tags, radio tags, in order to tag them and go out and continue to make observations, right? And so we can do like a mark and recapture. So if you put a collar on a lion or a tiger, and then you continue to search for tigers, you can like get the number of how many tigers you collared versus how many times you see those collared tigers again, and you see tigers that are not collared, and you can get an estimate of population size that way. They do camera traps and stuff like that. Camera traps is typically easier and less expensive than building collars, but that just kind of gives you an idea of how you could potentially do that. For your smaller rodent species, or they don't even have to be rodents, they could just be any smaller vertebrate. You can capture and mark those individuals. You release them, you go back and you recapture those individuals, and then you count the number marked that you recatch. And then so you combine the number with your first catch to the number in your second catch with the total mark. And so let me just kind of give you an example, right? So my previous lab that I graduated from at UC Merced, we go on the UC Merced Vernal Pools Reserve behind campus, and we will trap for small mammals because we want to get an idea of the small mammal population behind campus. And so what we do is we do some mark recapture data, and we gather that data so we can get an estimate of population size for the species. And now whenever we mark these species, so here you can see they put this blue X right on this individual. Now that will wash off, but the key is that it won't wash off within a couple of days, right? And so this sampling period is going to happen over 2, 3, 4 days, right? You're gonna trap for consecutive days. And so what we do is we'll go out and we'll take ear snips. So we'll take a little bit of tissue off their ear because we use that for DNA, so we can get information on population genetics. And so let's say we go out on the first day, right? We set our traps, we go back the next morning, and we capture 80 individuals, right? Let's say we capture 80 individuals. We take all of our data, we take our ear snips, and so those ear snips serve as a mark, right? So those 80 individuals are marked, okay? So that would be the number marked for the first catch. So we mark 80 individuals in that first catch, okay? So that's day 1, right? Then we reset the traps that evening, and then we go back on day 2, okay? And let's say we've caught 100 individuals, okay? So the day 2, so the total number in our second catch is 100, okay? Out of those 100 individuals, we look at every one of their ears, and we see if they were snipped from the previous day. And if they were, we know that we recaught them on the first day, and we basically recaught the same individual. And so out of our 100 individuals, 20 of them were marked. That means that 80 of those individuals are new individuals. They weren't marked, right? And so now knowing these numbers, we can extrapolate, right, based off of this formula to total population size. This is what this formula is letting us do based off of those ratios. And so the denominator is the number marked in the second catch. So we saw 20 marked individuals in our second catch, right? And so you just do the math. So 80 times 100, which is 8. Oh man, what is that? 8,000? Yeah, man, math is hard. Yep, 8,000. Then you divide 8,000 by 20, right? And so that would give you a total of 400 individuals estimated for that population size. Okay, and so that's how you can do mark and recapture studies, right? This number may be a little off, may not be the true number, but it's a good estimate, right? It gives us an idea of how large that population is, which is very important for conservation metrics and like whether deciding whether or not you can harvest that species. So for example, it's good to have this data for fish, especially if it's like commercial fisheries for large game that's hunted. So like deer, elk, you want to know exactly how large your populations are so you can put limits on how many can be harvested so that population stays healthy. Now, these are just two methods in which we can sample population sizes. There are many other ways you can do this. So for example, there's one that's called a line transect, where instead of doing the grid, the quadrat here, you would just you would have a line, right, that would go through the habitat, and you would walk that line, and you would stand on that line, and any organism that you could see that was part of the species that you wanted to count, you would count it. And then you would know the distance of that line and the distance you could see and the number you counted, and you can extrapolate that to a known size. And so you can get population size and density that way. And there are, again, many more other than that. Okay. And so that's how you can, or that is some ways you can determine population size and population density. Next, we can look at the distribution patterns of individuals within a habitat. And this distribution pattern is really important because it allows us to get information of how those species may interact with one another. So for example, the three distribution patterns we see is random, clumped, and uniform, right? Random is a pattern where, think about how seed dispersal is, right? Your seeds can get blown in the wind and they just randomly land in spots, and so your flowers are going to grow in these random patterns, right? So there's no real reason as to those growth or distribution patterns. Clumped typically will give you a few indications. One, maybe they're surviving in an area where all of the resources are, right? So for example, you're only finding flowers or organisms in one particular area, and all the areas in between that are bare with no individuals, and so maybe that's where the resources are located. Or it could suggest that they are a gregarious species like elephants, and all of your organisms are kind of clumped together in that distribution. Uniform kind of indicates a different interesting relationship. Uniform, you can think of like your almond orchards and that sort of thing. It's how you uniformly have them planted across the landscape. Now with uniform, you kind of see this in plants where they'll have like chemicals that their roots will release. And so basically those chemicals will be released in the roots, and they'll set out an area where that chemical prevents any other plant from living in that area, right? And so if each of these plants has its own area around it that no other plant can live in, right, you can see how that becomes spaced, right? How that becomes patterned. Or it could be nesting sites like you see here. right? It's particular nesting areas of maybe these penguins, right? So this penguin doesn't go in their nesting area and this penguin doesn't go in their nesting area. So you kind of have this uniform relationship. So it can really tell you a lot in how these populations are interacting with one another across the landscape. All right. So the next thing we're going to look at is basically the information about the life history of an organism and the life expectancy of individuals at different age classes. And so, excuse me, age classes can be defined pretty much any way you want to define them, right? So like maybe it's a plant that you categorize every age class every 3 months. So like an age class of 1 would be a grass individual that's between 0 and 3 months. An age class of 2 would be between 4 and 6 months. An age class of 3 would be between 7 and 9 months, right? Or you can have it based off of years. It could be 1 year old, 2 year old, 3 year old, or any kind of setup that you have dividing those age classes, right? And so you'll have those age classes and you'll want to know basically the mortality rates, the survival rates of individuals in that age class and the chance that they basically have to live and survive to the next age class. And so you can determine mortality rates by just dividing the number of individuals that die over the number of individuals that survive, and you multiply that by 1,000, and that gives you your mortality rates. Right? Actually, this should be 100. Right? So it turns it into your percentage. So there you go. So right. So if you have say 90 over 100, right, that's 0.9, or that's a 90%, right? That means you have a 90% mortality rate, which is not good for that population, right? But so it's number of individuals dying over number of individuals surviving multiplied by 100, right? Just to change it to a percentage. So that's not 1,000. And then, so for example, here we have a life history table of Dall mountain sheep. Okay. And so here you have age, and so this is years. And so you have basically from newborn to half a year to 1 year, 1 to 2, 2 to 3, 3 to 4, etc. And then you have the number dying in age out of every 1,000 that's born. So from half a year to 1 year, your mortality rate is a little higher than a newborn, and it's much higher than if you make it to a 1-year-old or a 2-year-old, right? And so you can see that mortality rate per 1,000 alive at the beginning of each interval. And so that mortality rate is super high for that age class of half a year to 1-year-old. So you know if that individual makes it to 1 years old, then they're going to be doing pretty well, right? So they'll be pretty good for the next couple of years, and then as they get older in age, the mortality rate increases significantly. All right, and so they have multiplied by 1,000 here in this formula to basically get these per 1,000. But a lot of the times, and actually it's more simpler to read, I think, if you just turn this into a percentage, right? If you just instead divide and then multiply by 100, and it gives you basically your percent mortality rate. So basically, if you have 937.5 per 1,000 dying, that's a 93% mortality rate, right? It's basically what you're showing. And then 1,000% mortality rate, right, is, basically, or 1,000 per 1,000 is basically a 0. You really don't see anything older than 14. Right. And then here you have your life expectancy, your mean lifetime remaining to those attaining that age. Right. So for example, if you attain 1 to 2 years old, your life expectancy is about 7 years. Right. So you're going to make it to about right here. Right. But this, again, this time frame here is kind of the key. If they don't make it past that, they're not going to do well. So this is kind of the key time in their life. Now we can take the information from each age class and we can turn it into a curve. So we can look at mortality rates across those age classes. For example, this Dall sheep has low mortality rates at a young age, right? So you can see here, and then higher mortality rates as an older age. Okay, so that would be a type 1 survivorship curve. Mortality is low in early years and middle years, and mostly occurs in your older individuals. So for example, humans, they have few offspring and they provide parental care because they have few offspring and they can put the energy into providing that parental care. For type 3 is basically the opposite of type 1. You have really high mortality as in early years, low mortality rates for older years. So here, in order to compensate for that more high mortality rate, they have lots of offspring, so thousands of eggs, thousands of seeds. And so if only 10% of their seeds survive and they have 1,000 seeds, then 100 individuals are going to make it, right? But because they're putting so much energy into offspring production, they can't provide parental care to their offspring because so many is there. They just don't have the energy and the capacity to do so. Right? Then you have your type 2 more, and so type 3 is things like plants, trees, and also some fish species are type 3, where they just kind of broadcast eggs or have a lot of eggs, and then those fish will hatch and have high mortality rates. But as long as they make it to an adult, they'll be fine. They'll survive to pretty much make it throughout their age classes. Whereas type 2, mortality is constant throughout the lifespan. So there's pretty much a constant mortality rate. And then an example of that is birds. Right? So here you have the different survivorship curves. So you have type 1, low mortality rate, young years, high mortality rate, older years. Opposite for type 3, so high mortality rate for younger years, low mortality rate for older, and then type 2 is constant across that time span, okay? Constant across age classes. Okay, so that does it for this first video for the module. We've talked about ecology, we've talked about kind of the concepts and parts of ecology. We refreshed on what a population is. We talked about how you can quantify and characterize populations, how you count species, how you determine density measures, life history tables, and survivorship curves. And so what we're going to look at now, or in the next video, is population growth and regulation. Right? So we're going to look at how we can model population growth through time. All right, and so we'll see you in the next video.